Welcome to this crash course on Alzheimer's disease. Before I start, I want to talk about a related term, and that is dementia. Most of the people here will have heard the term dementia and Alzheimer's disease, and most of us will even use it interchangeably. But it's important to understand the distinction between dementia and Alzheimer's disease. So I'm first going to start by telling you some horrible statistics about dementia. So 1 in 2 of us will see our lives devastated by dementia. And statistics carried out by Alzheimer's Research UK, which is a charity here in the UK, have shown that out of every 100 people in the UK, 55 of us will be impacted by dementia. That will be either by developing the disease ourselves or caring for a loved one that has it, or 11% of us will be extra unlucky and care for someone that has the disease and then go on to develop dementia ourselves. This is not just a local problem, it's a global one. So there are currently estimated to be about 55 million cases of dementia worldwide, and just under 1 million of those are here in the UK. Dementia is the biggest killer in the UK. Numbers are projected to keep rising. So these graphs here show the number of cases and how they rise in every year. And it's projected that over the next 25 years, the number of cases of dementia are going to double. You may notice that these graphs are separated into males in blue and females in red, and it's true that females are more prone to developing the disease, but the rise is the same from both males and females. So why is this happening? Well, one of the reasons is due to an increase in population. Year on year, there are more people in the world. And the other one is an increase in lifespan. We're very good at, medical advances means that we're very good at keeping people alive, and we're living longer life, but that does not correlate with an increase in health span. So even though we're living longer, it doesn't necessarily mean that we're staying healthy for longer. And the longer we live, the more prone we are to developing age-related disorders, such as dementia, and age is still the biggest risk factor for dementia. Now that I've given these statistics about dementia, we can move on to what I'm going to talk about today. So I will start by clarifying the difference between dementia and Alzheimer's disease. I'll then move on to the actual crash course in Alzheimer's disease, talking about the history of the disease and what we currently know about it. Towards the middle, I will start talking about how we study the brain and the research that we carry out here in the Cellular Phase of Alzheimer's Disease Lab. And towards the end of the talk, I will touch upon the future and what that holds for patients and their families. So let's get straight to it, dementia and Alzheimer's disease. Well, even though, as I said, most people use the terms interchangeably, dementia is not Alzheimer's disease, and it's not even a disease. Rather, it's an umbrella term used to describe a collection of brain diseases and their symptoms. And these include memory loss, impaired judgment, personality changes, and an inability to perform daily activities. Dementia can arise from various underlying diseases. One of them is Alzheimer's disease, the main focus of our talk today. But there are also other dementias, such as vascular dementia, so that will be following a stroke or a brain bleed, frontotemporal dementia, Lewy body dementia, and other dementias related to other diseases such as Huntington's disease, Parkinson's disease, or even from traumatic brain injury. So we will all have heard in the news stories from ex-footballers, ex-rugby players who have gone on to develop early symptoms of dementia following injuries that were sustained during their professional careers. Alzheimer's disease is the most common cause of dementia, and it accounts for 60 to 80% of all cases. It's a very specific disease, and it's characterized by the presence of amyloid plaques and neurofibrillary tangles in the brain, and I will talk a little bit more about those later on. In Alzheimer's disease, which we can shorten down to AD, neurons, which are the excitatory cells of the brain, they degenerate and die, and this causes the brain to shrink. And in fact, it's calculated that over the time course of experiencing dementia, you may lose about an orange worth of brain matter. So that's quite a significant amount. And changes in the brain can start decades before clinical symptoms, and this poses a complication when it comes to studying dementia. So not only does it affect the most complicated organ in the body, the brain, but also we now know that changes are happening in the brain way before the symptoms begin. And therefore, how can we start treating someone when they come to the clinic? It'll be too late. Changes have been happening for years and even decades. So we need to divide research into Alzheimer's disease into 3 stages. 1, we need to find ways of early detection 2, we need to understand the disease, what's happening in that time between the first changes that are happening and the dementia symptoms. And 3, obviously, we need to develop therapeutics for patients and find support for their families. In this picture here, which I haven't mentioned, you have a representation of what happens to the brain in dementia. So on the left, you have a normal brain, and on the right, you have a picture depicting what would happen in Alzheimer's disease. So you can see that it's represented as a smaller brain, and this area here that's highlighted by the black box is called the hippocampus, and this is a center for learning and memory, and it's particularly affected in Alzheimer's disease, and we think that it's because of this that we get the symptoms of memory loss. So there are still many unknowns about Alzheimer's disease. The history stretches back almost 120 years. So back in 1906, the amyloid plaques, which are defining characteristic of Alzheimer's disease, were first identified. This was done by 2 scientists, Alois Alzheimer. It's after him that the disease was named. He was a German psychiatrist, and he encountered a patient, August Dieter, who came to his clinic, to his psychiatric hospital, aged 51. This was very early on for any patient to develop symptoms of dementia, and she was experiencing memory loss, she felt disorientated, and she described herself as losing her mind. Alois Alzheimer had many conversations with August over 6 years, and then she passed away at the age of 57. After she died, Alois Alzheimer took out her brain and used novel staining techniques at the time to look at what was happening in her brain. And he identified these amyloid plaques and neurofibrillary tangles. At the same time, Oskar Fischer, who was a Czech clinician, he also used the same novel staining techniques to look at the brains of senile patients. And he also discovered these plaques and reported them. These plaques are still what we use today to identify Alzheimer's disease. So we have the amyloid plaques. They are abnormal clumps of protein that are seen spread across the brain. And neurofibrillary tangles are clumps of a different type of protein, in this case a protein called tau, and they're found mainly clumped inside the neurons. Now, I'm not going to touch much about tau today, but it is important to know that it's this clumping inside the neurons which is thought to lead to their death. And also, when neurons have this abnormal tau, it's become phosphorylated. They can shed the tau, and it's this action that is believed to be part of what spreads the disease. When the neurons start dying, they start showing these granules and vacuoles inside the neurons, and this indicates a specific method of cell death called granulovacular degeneration. And to this day, 120 years after the first discovery of the signs of Alzheimer's disease in a brain, we still use the same techniques. We stain brains from post-mortem brain and can definitely say if someone's had Alzheimer's disease. Now, you may have noticed that I keep saying that we do this in post-mortem brain, and that is not helpful to a patient. It is not helpful to any patient to be diagnosed with the disease after they're dead. Thankfully, technologies have advanced, and we can now use PET imaging in the live brain in humans to look at accumulation of that protein that I keep talking about that forms the plaques, amyloid. So we have on the left a healthy control, and the cold colors, blue, green, purple, show low levels of amyloid. In an Alzheimer's disease patient, you see hot colors, yellow, orange, red, and they indicate high levels of amyloid building up. This technology is great, and it can really save someone's got Alzheimer's disease, but the problem is that this is really expensive, and it's very limited. So the reality is that less than 2% of patients will get this test and have a definitive diagnosis during their lives. And unfortunately, the stark reality is that most patients won't actually be diagnosed officially until after they're dead. Alzheimer's disease can be categorized into 2 main forms: early onset Alzheimer's disease and late onset Alzheimer's disease. Early onset relates to any patient that shows clinical signs of the disease of dementia before they're 65 years old. And in late onset, most patients tend to develop Alzheimer's disease in their 70s or 80s. Early onset, fortunately, only accounts for 5 to 10% of cases, and a large proportion of those cases can be further categorized into familial Alzheimer's disease. So this will be the case when Alzheimer's disease is passed on through generations within a family. And although this accounts for less than 5% of all cases of Alzheimer's disease, it's through studying these patients with familial Alzheimer's disease that we have made major advances in our understanding of the disease. Okay, so what do we understand about the disease? Well, initially, I said 1906, we discovered amyloid plaques. We didn't know what they were made of. We didn't know if they were cause or consequence of the disease. They were just an observation in a patient's brain. And not much happened until the late 60s, mainly because we didn't have the technologies. In the late 60s, the main component of plaques was discovered to be a protein called amyloid. But it wasn't until the late 80s, early 90s, that the gene responsible for this protein was discovered, and that was APP, this one here. And APP stands for amyloid precursor protein. So what we now know is that amyloid precursor protein is a large protein that is expressed mainly in neurons in the brain, and to perform its actions, it needs to be chopped up into several fragments. One of those fragments is called amyloid beta. When amyloid beta is made, it usually carries out its action, and then it gets cleared away from the brain by specialized cells called microglia, and I will be talking more about microglia later on. But amyloid beta can come in various sizes, and in patients that have a mutation in the APP gene, what happens is that that fragment is actually longer than in healthy patients. And when it's longer, it starts misfolding. And so when it misfolds, it starts clumping together to form what's known as a toxic oligomer. The toxic oligomers then clump further to make fibrils, and the fibrils clump together to make amyloid plaques. It's not just mutations in APP that can lead to Alzheimer's disease, but also in a couple of genes called presenilins, presenilin 1 and 2. And they code for the enzyme that is responsible for cutting up amyloid precursor protein. So again, if there's a mutation in these genes, the chopping up of the precursor protein goes a bit wrong, and then this leads to the clumping of amyloid. So this led to the amyloid hypothesis in 1991. And this hypothesized that you have your mutations in APP, you then get a clumping of the amyloid, this leads to the plaques, and somehow, by an unknown mechanism, that leads to the neurofibrillary tangles in neurons, and ultimately cell death. It's very linear, it's a very neuron-centric hypothesis, and it's shaped much of the research that has happened ever since, including clinical trials. Now, we now know that it's not quite so simple. Many things are happening in the brain, and it's not just related to neurons. So we have this complicated picture that I've drawn here. The neurons are shown in orange, and they start degenerating. But there are other cells in the brain, those glia that I talked about, and other vascular cells, and they've all been shown to play a role in Alzheimer's disease. And so it's by joining up all these little pieces of the puzzle that we start to understand more about the disease. And so while the amyloid hypothesis remains a cornerstone of Alzheimer's disease research, it is clear that the disease uses up many more cells in the brain, and we need to understand all the cellular interactions to try and understand more about the disease. If we look at the genetics, as I said, a lot of research and milestones in Alzheimer's disease research have been based on genetics. I mentioned already the APP and presenilin genes. Mutations in these genes will lead to getting Alzheimer's disease, and these are called causative variants. The reality is that familial Alzheimer's disease accounts for less than 5% of cases. And so what is it about the rest of the people that are getting the disease that's causing them to get Alzheimer's disease? Well, at the time, it was very difficult to sequence someone's genome. It took many years to just find these genes. But nowadays, we can look at the genome of many patients in one go. And studies have started to reveal other genes that may play a role in Alzheimer's disease. And these are shown here. So in this complicated graph, the higher they are, and then shifted to the left, the higher the chances of you developing Alzheimer's disease. So as I said, these are the causative variants. Mutations in these genes will lead you to have the disease. Whereas on this side, these genes have been shown to increase your risk of developing Alzheimer's disease, but not necessarily cause Alzheimer's disease. And there's a large number of genes. Anything above this line means that your risk is increased, and anything below it means that your risk is decreased. And what's interesting about these genes is that a large proportion of them are not normally expressed in neurons. In fact, many of these genes are expressed in microglia, which, as I said, are important for clearing away amyloid beta. And this one here, ApoE4, which is one of the ones that has the highest risk, even though it's not causative, is expressed in astrocytes, which is another cell type in the brain. So I will talk a bit more about those later. When it comes to causes of the disease, and I say causes in inverted commas because, as I said, most of what we know is about increase in risk or decrease in risk, but not necessarily causative. We've talked about the genetics. I've also mentioned that aging is the highest risk factor, so the older you get, the more chances of developing the disease. And then there's about 40% of risk is by potentially modifiable environmental and lifestyle factors. And this includes things like your level of education, hearing loss, how much you drink, smoke, depression, social isolation. All these are shown to change your risk towards Alzheimer's disease. And we don't know how this happens. And I'm not saying that if you were to mitigate these risks, you won't get the disease, but we always say that what's good for your heart tends to be good for your brain, and it makes sense to try and keep these to a minimum. As we stand at the moment, we have many pieces of information that we are all trying to piece together to fill this jigsaw puzzle on how Alzheimer's disease comes to be and how the progression occurs. As we've learned more about the brain, we've learned more about Alzheimer's disease, and if we look at the number of publications, they have been increasing steadily over the last 50 years. So this is knowledge for us. The more publications, the more we know about the disease. Alzheimer's and dementia researchers, we like to compare ourselves with cancer researchers, many of whom work in this building. And if we look at cancer, the knowledge that we have on cancer is far, far greater than what we have on Alzheimer's. So this is still the same pink curve. The axis has now changed. And these are the number of papers published in cancer. We have a lot of knowledge in cancer, and I'm not trying to make unfair comparisons between the two diseases. What I want to draw your attention is that knowledge is power. The more we know about the disease, about every single aspect, we will know more, and we can tackle it. 20 years ago, if you went to the doctor and you were told you had cancer, that was a death sentence. And now, through efforts from scientists all over the world and in this building, we've learned more about the disease, and we've developed therapeutics so that now some cancers are even curable, and if not treatable. That's what we strive for in dementia research, and we're hoping that the more information that's coming along will help us find this cure for Alzheimer's disease. So with that, we can move on to the next part of the talk, which will be about how we research mechanisms of disease and what we're doing here in the cellular face of Alzheimer's disease. Okay, so why are we called the cellular face of Alzheimer's disease? This was actually a term coined by my colleague, Bart de Strooper, with whom I run the lab, and his collaborator, Eric Caron. And they introduced this term back in 2016. So I've already mentioned that many years before you have clinical symptoms of dementia, there are changes happening in your brain. And these are depicted by these curves here. So the first thing we see is an increase in amyloid beta levels in the brain. This is followed by an increase in that other protein, tau. And then you see changes in the brain structure. So if patients that will have had MRI scans, regular MRI scans will show that their brain is shrinking. Following that, you start seeing the memory loss. And beyond that, the other clinical symptoms of dementia. So we know that there's this biochemical phase when the amyloid is being built up, and we know that there's the clinical phase when a patient goes into the doctor and seeks help. And what Eric and Bart hypothesized is that in this interim, it's how the cells are responding to these increasing levels of amyloid and tau that drive the chances of you developing the disease. So we really need to understand what's happening to the cells at this stage to understand why we're getting the symptoms of dementia. And it's important to note that actually, if you look at post-mortem brain tissue, many patients will have lived until the 90s, even centenarians, and if they are kind enough to donate their brain, some of them, when we do the stainings, we'll see that they actually have high levels of amyloid plaques. Now, they have high levels of amyloid plaques, but they didn't have any symptoms of dementia. So what was it about those patients that meant that they didn't develop the disease, whereas other patients go on to develop clinical signs of dementia? So how do we study the brain? Well, there are many different ways. I've already mentioned about the genetics by looking at DNA that's found in all the cells in our body. And I've also spoken about the progress that's been made in looking at changes in the patient during their lifetime and afterwards in post-mortem tissue. But there are other ways that we can investigate what's happening. One of them is by looking at RNA expression. And so all the cells in our body have DNA, and they all have the same DNA. But a nerve cell is different to a muscle cell, which is different to a skin cell. And that depends on which genes are being chosen to be expressed. So think of it as a library. You go into the British Library, and there are many books. Every cell has access to the same books. But it might be that one cell prefers reading fiction, whereas the other one prefers reading true crime. So that's how we look at genetic expression. And we can infer a lot from the RNA, because that is what genes are being expressed, so which books are being chosen, just like you can infer a lot from what someone reads. That will only tell us part of the story. RNA is then used to make proteins, and these are the functional building blocks of our cells. So it's also important to look at what proteins are being expressed and how these proteins are changing over time as the disease progresses. It's also important to look at cells as a whole. So we look at their activity, their function, their behavior, and how they communicate with each other, be it in vitro or in whole brains. And so really overall, our research uses a holistic approach, combining various methodologies to understand what's happening in Alzheimer's disease and uncover novel insights into the disease. There are many different types of cells in the brain, as I mentioned. Neurons are the most known. They are the excitable cells of the brain, and they communicate with each other. They come in various shapes and sizes, and if you go into the Manby Gallery after this talk, you can go into the Hello Brain exhibition, and you will see that we have models of different brain cells, nerve cells, that had been crocheted, and they come in all these funny shapes, and you can learn more about how they communicate and how they shape brain function. This is an example of two hippocampal pyramidal cells. They come from that area of the brain that I said was particularly affected in Alzheimer's disease. And they communicate at these specialized junctions called synapses. So this is just a diagram here about how one cell can have a little protrusion go into one of these little things that stick out from the neurons. That's a spine. And this is how they communicate. And this is an electron micrograph to show what they look like in real life. Now, these are the cells that degenerate in Alzheimer's disease, but they're not the only cells found in the brain. Other cells are microglia, and I mentioned that these are important for clearing away amyloid beta. They do a large number of things in the brain. They're very important cells. They're the immune cells of the brain, and they're constantly striving to protect our brain. They move around, and I'll show you a video of them later moving. And if they detect any damage to the brain, they will collectively move towards that region, patch it all up, and make sure that the damage isn't spread any further. They also clear up debris, like amyloid beta after it's been used, or if a cell were to reach the end of its lifespan and die, microglia will go over and vacuum that up. They're also important for a process called synaptic pruning, and this occurs in the transition from childhood to adulthood. So I said that spines are part of this specialized junction, the synapse, and actually it's thought that these junctions hold our memory. So when you teach a task to a rodent, once it's learned that task, scientists have shown that they have more spines and bigger synapses. And in the development from childhood to adulthood, we need to get rid of some of these synapses to leave space for new neuronal networks to be formed. And this is called synaptic pruning, and microglia play a critical role in this stage. And then the other thing that they do is that they release nice chemicals to keep the brain happy in a healthy environment. In disease, microglia are important in the inflammatory response. And like inflammation in any other part of our body, microglia it can start off as a good thing, but if it moves on to chronic inflammation, then it starts being detrimental. And microglia can start releasing pro-inflammatory cytokines that may start damaging other cells. In Alzheimer's disease, it's been shown also that they start aberrantly phagocytosing synapses. So those pathways that are important in that development stage from childhood to adulthood, where they strip those synapses, that's in a controlled way. In disease, microglia have been shown to start activating the same pathways, but in a situation where we don't want that to happen. And that is thought to be correlated with that memory loss that we see. They can also break down something called myelin, which is an important part of our brain. It wraps around neurons, and it's important for the transmission from one cell to the other. And in disease, what also we see is that they change their shape. So microglia, as I said, they're shaped like this, and they move around. And what we see is that they change their shape, they become more blobby, and they stop looking for any injury in the brain. And obviously, that is a problem. The cell types that we study are astrocytes. These are the most abundant cell type in the brain. They account for 20 to 40% of brain cells. And originally, they were thought to have very little function. They were just thought to be scaffolds in the brain. They were thought to be the glue, and that's what actually how the term glia came about. It came from the word glue. Turns out that astrocytes are way more interesting than that. They're important for various functions. They're up there at the synapse as well, modulating activity between neurons. They keep the brain in homeostasis. That means that it keeps it happy. They interact with the vasculature, and they are also responsible for the inflammatory response. And as I said earlier on, astrocytes are the cells that express that risk gene, ApoE, that significantly increases our chances of developing Alzheimer's disease. Now, Disha in our lab has also shown that that astrocytes in Alzheimer's are one of the first responders to increasing levels of amyloid. So even before plaques are formed, astrocytes start changing their behavior. The importance of these two cell types, I hope, is depicted in this picture here. This is one of the first pictures that I took when I started working on Alzheimer's disease. And I just stained a brain section from a mouse model of Alzheimer's. And in red, you can see the amyloid plaques. So you can see that the amyloid really clumps up into these plaques. In blue, I have labeled astrocytes, and in green, microglia. They're beautiful cells. And hopefully you can see that at the plaque, there's a concentration of these cells. These cells migrate to amyloid plaques and work together to do something. What that is, we still don't know. So what we're interested in the lab is to try and identify the communication pathways between astrocytes and microglia and neurons, and how they respond to increasing levels of amyloid. Okay, so what are we doing in the lab? One of the things that we're looking at is how male and female microglia have different responses to amyloid. So I said at the beginning that females are more likely to develop the disease. Now, the reality is that males, when they develop the disease, they have shorter lifespans and more severe symptoms. So we're trying to understand what's different about male and females and what's driving these differences on how they respond to the disease. We've hypothesized that perhaps the responses from microglia may be different, and we've teamed up with James Turner, who leads the sex chromosome biology lab here at the Crick, to ask this question. So I mentioned that one of the things that we look at is RNA expression, and I said that that's which genes are being activated in particular cells. And microglia actually come in various different cell states. So even though they're the same type of cell, they may express different genes. So see it as, okay, you have a group of people that like to read fiction. That's our microglia. They all like to read fiction, but some of them may be interested in magical fiction, science fiction, or crime fiction. So they're separated into different subgroups. And we find that these cells, which are represented here in different blobs, they change their preferences as the disease develops. We've been looking at these responses in males and females, and we're comparing in this graph here, the males are on the left and the females on the right, and each dot here represents a different gene. And we can see that microglia from males and females don't express the same genes because they're not necessarily aligned on this vertical line here. And the further away they are from this vertical line, the more different they are. And so we're now looking at this particular blob of blue genes here to try and see if this will give us information about why there's a difference between the male and female response. When we do these experiments, what we do is we take a piece of the brain and we mush it up, isolate the cells, and we can ask what genes are being expressed in individual cells. But it doesn't tell us much about where the cells originated from. They just come from our chunk of brain. Technology has now moved on to allow us to ask that question as well. So not only what is being expressed in different cells, but where are those cells in the brain? And this is called spatial transcriptomics. When we do this, this is a pictogram. Well, these are actual results here, showing how if we separate cells based on which genes they express, you can see that they are found in different parts of the hippocampus. This is a mouse hippocampus, and it's a great piece of the brain to study because it has a very defined architecture, and different cells are found in different areas, and they are highlighted here. So this shows that the technique works. We can then start asking, well, what are the genes that are being expressed closer to amyloid plaques, which are depicted here in white, and far away from plaques? So do all microglia, do all astrocytes, neurons in the brain react in the same way, or do they behave differently depending on how close they are to plaques? And Anna and Magda in the lab have shown that there are a number of pathways that are correlated and upregulated near plaques, and they are related to communication between astrocytes and microglia and neurons. I told you that RNA is only part of the story, so we're also doing similar experiments asking what proteins are being expressed by these microglia near plaques and away from plaques. And to do that, we do a slightly different experiment. What we have is a very cool microscope upstairs that can cut out individual cells and put them in a tube that can then be analyzed for proteomic analysis. And proteomics means that we get to look at many proteins expressed by a given cell. We can't do it by one individual cells just yet, but we pull them in groups of 100, which is actually a very small number. This is highly time-consuming, and it can be prone to bias. So Emma, what she's done is she's developed an algorithm using machine learning to detect the cells and separate them based on their shape and proximity to the amyloid plaques. And I'm not going to go into the details of the results here, but this heat map shows that there's different protein expressions in different types of cell types. And so by looking at this together with RNA analyses, we aim to unravel intricate molecular mechanisms underlying Alzheimer's disease and identify potential therapeutic targets. If you haven't noticed, I'm very interested in microglia and astrocyte crosstalk. And I've already mentioned how we use these gene expression networks by looking at the RNA, but we also use other methods to look at what's going on there. So we use biochemical approaches combined with that proteomics approaches to identify molecules that are being released from different cell types in response to amyloid pathology. And we can do this over time, so we can actually see how this changes as amyloid increases. And importantly, we can also do in vivo functional imaging. So in a mouse model, we can actually look at their brain at different stages of the disease while the animal's still living and see how this cellular behavior changes over time. And so this is that promised video that I had of microglia. And you can see that they're moving around constantly. We can now image these cells together with astrocytes and look at astrocyte function. And we now even have the capability to modulate, to our will, astrocyte function and see how this affects microglia biology. So in essence, our ongoing research endeavors to seek to uncover the intricate relationship between these cell types and shed light on novel pathways that could hopefully then be used for therapeutics. Now, this was all very complicated, I understand that. So if there's only one message that I would like you to remember by the end of this talk, is that Alzheimer's disease progression is a complex multicellular process. I've only spoken about 3 different cell types, neurons, astrocytes, and microglia, but the reality is that we also know that other cell types, such as oligodendrocytes and pericytes here, are also important and contribute to disease pathology. With that, we're coming to the end of the talk, so I want to touch on what does the future look like? And really, when I mean the future, I actually mean the present, because if we're in a really exciting time in Alzheimer's disease research, and if you've been keeping an eye on the news, you will have seen numerous headlines over the last few years that keep propping up on Alzheimer's therapeutics. So 3 years ago, the US approved the first new Alzheimer's drug in 20 years. This drug is available in the US, it's not available in the UK or Europe, but it really was a turning point in Alzheimer's disease therapeutics, and it offered hope to patients and their families. Since then, there's been 2 other new drugs, lecanemab and donanemab. They work in similar ways, and really it's just providing this hope and this idea that we may be able to tackle Alzheimer's disease. And there's a summary about these drugs in a recent Panorama episode that was aired just 2 weeks ago. I really recommend watching it. It's only 29 minutes long, and it can tell you all about the clinical trials that surrounded these drugs. These drugs are based on the amyloid hypothesis. So as I said, the amyloid hypothesis has now been shown to be way more complicated than originally thought, but we still believe that amyloid may be the trigger of the disease. And so these drugs, what they do is that they're an antibody, all of them are antibodies that bind amyloid in various different stages of its clumping, and then allow for microglia to come and take that amyloid away in an easier way. And if we look at the results from the drug companies, on the left you have the results from lecanemab. You can see that PET scan from a patient at the start of the study. It has the hot colors, really high levels of amyloid. And after a year of treatment, they show that the amyloid levels in the brain have been significantly reduced. Donanemab, a different drug, but also an antibody to amyloid. If you look at a clinical way that doctors have of measuring cognitive decline, and that's how it happens when you have the placebo. And those people taking the drug, you will hopefully see that the decline is not as strong as in the placebo. But you will notice that the decline is still there. It's just shifted. It's delayed, but it's still happening. And this really reiterates the point that I've been trying to make, that it's not just about amyloid buildup. We need to get rid of the amyloid, that's for sure, but we also need to tackle those cellular responses that are happening, because once they get started, they're happening in parallel to amyloid buildup rather than because of the amyloid buildup. So these drugs, they're great, they're offering hope, but to be on these trials, you have to have this magical diagnosis, which I said only 2% of patients get. And 10 days ago, this big headline was in The Guardian, thousands to miss out on these new dementia drugs. So we're at this fine balance where we have potential drugs that may help, but we don't have the capacity to test patients to see if they're eligible for the drugs. Hope is not lost. They've now shown that phosphorylated tau, which is that protein that builds up and forms the neurofibrillary tangles, can be detected in blood plasma. And that was a paper published in Nature Medicine. And you will have seen in the last week these headlines about how new trials are starting. Thousands are to trial blood tests for dementia. How great would it be if you can go to a doctor and just get a blood test, which is relatively painless, very quick, and be told if you're going to develop dementia. Even better, if there were a therapy that can offer you hope to treat it. Last year, I was at a talk by John Hardy, now Sir John Hardy, who first came up with the amyloid hypothesis, and he likened these advancements as the first flight in North Carolina by the Wright brothers. It's a scary plane, and these are scary drugs. They have huge adverse effects. But it was a turning point. After this flight, we had the first transatlantic flight, and now there are hundreds of flights leaving airports every day to get people closer to their loved ones, go on holiday or a conference. And we really think that this might be a turning point for Alzheimer's disease, and soon we'll be able to develop new therapies and offer hope to patients and their families. So just to finish, I want to thank Bart de Strooper, who gave me the opportunity to set up the lab with him here at the Crick, the lab who worked tirelessly on all the projects that I've shown you, our collaborators, and of course the technology platforms here, advanced sequencing, light microscopy, and the BRF, without whom we wouldn't be able to do any of the work that I have presented. So with that, thank you very much. and I'll take any questions.